The ability to communicate with one another is important to the way cells function. It's a common feature of single-celled prokaryotes and at all multicellular organisms. In this video, you'll learn about how signals are sent and received and how those signals bring about changes in their target cell. Communication between cells is very much like communication between individuals. In most cases, there's a sender who sends a message that is received by a recipient or a target. The message that the sender sends travels between the individuals and is received by some sort of sensor on the target. In this case, the sensor is highly attuned to receiving sound waves, but is incapable of receiving other forms of communication. Once the signal has reached the receptor, it gets passed along a sequence of pathways until it can cause some sort of response or change in the target. We'll start by looking at how the distance between the sender and target affects the nature of the message. If you're touching somebody, you can send a message by squeezing or rubbing or whispering very softly. If you aren't touching, but are still in close proximity, then these tactile methods won't work, and you send a message by talking or maybe a visual message with your expressions. When you're very distant, perhaps halfway across the country, you can communicate by email, phone, text, etc., but not by shouting or anything tactile or visual. In a similar way, the distance between cells makes a difference in how they communicate with each other. At the very local scale, cells are in direct contact with each other and are able to pass signals directly between them. Many cells are able to communicate over very short distances, usually sending molecules directly from one cell to another. This direct communication exists in prokaryotes as well as multicellular organisms. At a much larger scale, signals are able to be sent and received from one part of a body to another very distant part. Let's see how this works. Remember when we learned about the cytoskeleton, that we learned about plasmodesmata in plants and gap junctions between animal cells. Both of these structures provide continuity between the two adjacent cells, such that the cells share cytoplasm and small molecules can readily move between the cells. This is a particularly effective way of signaling because the signals don't have to go through any cell membranes, since the plasmodesmata and gap junctions allow continuity of the cytoplasm. This type of communication is only useful for cells that are adjacent and have a long-term relationship with one another. Cells that are adjacent to each other on a transient basis don't form gap junctions or plasmodesmata. Cells that are more mobile may tend to communicate through cell recognition pathways. Proteins and other molecules on the surface of the cells can be identified by other cells, and binding to surface proteins may trigger a response in the recipient cell. This type of recognition is the primary mechanism by which cells in the immune system identify foreign invaders and prepare full-scale attacks on infectious agents. Signaling can, of course, happen between two cells that are in very close proximity to one another, even though they aren't in direct contact. Paracrine signaling involves the cell secreting signals, often proteins, via secretory vesicles into the extracellular matrix. These molecules travel short distances to target cells where they cause some change in that cell, in that target cell. Growth factors or cytokines are good examples of this type of communication. Cytokines are involved in cell signaling as part of the immune response. They're released at the points of infection and signal to white blood cells and other parts of the immune system where the infection is located. Another type of local signaling occurs between neurons across gap or the synapse that separates neuron cells. Neurotransmitters are released by the presynaptic neuron as a result of an electrical signal reaching the end of the neuron. neurotransmitters diffuse across that tiny synapse where they bind with receptors in the target neuron and can activate or repress the postsynaptic neuron from propagating the electrical signal. Direct contact signaling and local signaling are great ways for cells to communicate with one another. However, it requires that they're located adjacent or at least very close to each other. Cells that are a distance in a multicellular organism may communicate through long-distance signaling. In animals, this occurs through the endocrine system, in which endocrine cells produce hormones that are dumped into the bloodstream. The bloodstream carries the hormones throughout the body, and some of those hormones find their target cell, which has appropriate receptors on its surface. In plants, molecules may travel through plant vessels, diffuse between cells, or they may travel as a gas from one region of the plant to another. Let's look in general how messages are received and operated on by the recipient cell. This figure shows a generic pathway in which a signaling molecule binds to a receptor that's an integral protein of the cell membrane, and thus extends from the outer surface of the cell through the phospholipid bilayer and into the cytoplasm. Binding to the receptor causes a change in that integral membrane protein, and that change is felt inside the cell. Transduction passes the message through a series of other molecules until it can cause a response by the target cell. The response in the target cell may be in the cytoplasm or it may be in the nucleus. Let's start by looking at different ways in which a cell can receive a message. Cells receive chemical messages in two ways. They may receive the message at the cell membrane for chemical messengers that can't pass through the membrane. Some molecules, of course, can pass through the membrane, and these are often received by receptors inside the cytoplasm. Hydrophobic and/or small molecules are able to move through the membrane, and once inside the cytoplasm, they bind to a receptor protein. Once bound, the effects of the message may start. In the case of the hormone aldosterone, binding to the receptor activates the receptor and allows it to enter the nucleus. Once inside the nucleus, it acts as a transcription factor which binds to DNA and essentially turns on transcription of particular genes. Turning on a gene results in transcription to make more messenger RNA, and this messenger RNA passes out of the nucleus and is translated into a protein. Thus, the hormone has entered the cell and caused the cell to produce a protein that has some other effect. Notice that since these signal molecules can simply pass through the membrane, it's likely that the molecule will enter many cells rather than just the target cell. So, how does the organism control which cells should respond to such a message? Well, it does this by providing or not providing the receptor protein. If the cell is a target for this hormone, then it will have a receptor to which the hormone can bind. If it's not a target cell, it won't have that receptor, and the message essentially won't be heard by the cell and will therefore have no effect on that cell. Most cellular messages are not able to get into the target cell because they're hydrophilic or simply too large to enter through the membrane. In these cases, the messaging molecule is received at the cell surface by binding to a protein embedded in the membrane. When this happens, we call the signaling molecule a ligand, and it binds to the receptor protein, which is usually much larger. There are three main types of receptors: G-protein coupled receptors, or GPCRs, receptor tyrosine kinases, or RTKs, and ion channel receptors. Although these each work in their unique way, the effect is the same in that they receive the messaging molecule and then and then they cause some change to happen on the inside of the cell. G-protein coupled receptors are large proteins embedded in the plasma membrane. On the outside of the membrane, they have a binding site to receive particular ligands, and on the inside, they have a binding site for a G protein. The G protein can be classified as either being on or off, depending on whether it has a GDP or a GTP attached. Now, GDP and GTP are similar to ADP and ATP, except they're made of guanine instead of adenine. But they have a similar effect in terms of their energy delivery. When it's inactive, the G protein binds to the G protein coupled receptor. When the receptor receives a messaging ligand, a signaling molecule, it changes shape and causes the G protein to, on the inside, on the cytoplasmic side of the receptor, to expel its bound GDP. It then has space and the affinity for binding to GTP, and now that it's bound to GTP, it's considered to be turned on or active. The G protein can then dissociate from the receptor and move along the inside of the plasma membrane and bind to an enzyme. Once bound to the enzyme, it becomes and can cause further reactions involved in the transduction of the chemical signal inside the cell. The G protein is also a GTPase enzyme, which means that it hydrolyzes GTP into GDP. So once it activates the enzyme, it retains the GDP, but it's now inactive. This means that it's now able to receive another message from the receptor if the receptor becomes activated by another signaling molecule on the outside of the cell again. This activation by GTP allows the cell to shut down the signaling pathway as soon as the signaling molecules are no longer present. The second class of receptors are called receptor tyrosine kinases, or RTKs. Like with other receptor molecules, they bind a signaling molecule on the outside of the cell and cause some changes on the inside. As with kinases, as they are kinases, receptor tyrosine kinases are capable of transferring the phosphate group of an ATP onto a tyrosine residue and converting the ATP to ADP in the process. Receptor tyrosine kinases usually exist as single units or monomers. When a signaling molecule binds to them, however, two receptor tyrosine kinases associate together into a dimer. This dimerization activates the parts of the molecule that's inside the cell, and it activates it, allowing it to hydrolyze ATP and ADP into ADP, and in the process transfers the phosphate group onto the tyrosine residues. Once the tyrosine residues are phosphorylated, the receptor is active and can be recognized by relay molecules. These relay molecules take the phosphate and in turn become active themselves. Perhaps the biggest functional difference between G protein coupled receptors and receptor tyrosine kinases is that G protein coupled receptors usually only activate a single pathway inside the cell. Receptor tyrosine kinases, on the other hand, are able to activate a whole host of responses. Each of the tyrosine residues bound to the phosphate group may activate a different relay protein. Each of these, in turn, may set off a separate cellular response. As a result, the single signaling molecule on the outside of the cell can cause many pathways inside the cell to be activated all at the same time. The third type of receptors are ligand-gated ion channel receptors. Like with other receptors, they have a binding site on the extracellular side of the membrane. When a ligand binds to that site, it causes the receptor to open. When it's open, ions may flow through the channel down their concentrating their concentration gradient. This may involve ions flowing into the cell, or the ions may flow out of the cell, depending on the concentration gradient. The change in the concentration on the inside of the cell is what causes some responses by the cell. This type of ion channel is what allows neurons to communicate with each other. The region where two neurons meet is called the synapse, and it's a small gap between the neurons. When the end of the neuron that's sending the message receives the electrical signal called an action potential, this signal causes neurotransmitters to release by exocytosis. The neurotransmitters rapidly diffuse across the synapse, and some of them bind to a ligand-gated ion channel. This allows the influx of sodium ions. If enough sodium ions flow into the cell, perhaps because there's a very strong signal from the sending neuron, then the action potential, or the electrical signal, will start in the postsynaptic cell and be propagated wherever that cell leads. As we've seen, there are four primary ways of receiving a signal. The signal can be taken directly into the cell and received by a protein in the cytoplasm, or the message can be received at the cell membrane by either G-protein coupled receptors, receptor tyrosine kinases, or ion channel receptors. Once the message has been received by the receptor, it gets transduced or passed on in a multi-step pathway until it causes a response by the cell. One of these transduction pathways is called a phosphorylation cascade. Remember that proteins can be activated or deactivated by the addition of ATP. In a phosphorylation cascade, the activated receptor activates a relay molecule, and this in turn activates a protein kinase. Once activated, this protein kinase is able to attach phosphate groups onto other protein kinases. As one kinase phosphorylates another, which may phosphorylate another and another, this is known as a phosphorylation cascade. Eventually, an activated protein kinase phosphorylates a target protein, which can then cause a cellular response by the cell. It may seem that once all of those protein kinases are phosphorylated, there's no way to repeat the signal and message. This is almost like sending a message by writing the message on all of your paper. Then, when a second message needs to be received, there's no paper left to write the letter on. However, if you have an eraser, you can erase the first message and then use the paper to write the second message. Well, in a phosphorylation cascade, molecules called protein phosphatases help remove the phosphate groups from the proteins in the cascade, making them available again for the next message. A second important transduction pathway involves a molecule called cyclic AMP. This pathway is usually associated with the G-protein coupled receptors. As with other G-protein coupled receptor signaling, a ligand binds onto the GPCR, and this causes the G-protein to expel its GDP and accept a GTP molecule. The G-protein transfers the ATP to an enzyme and activates it. In the case of the cyclic AMP pathway, this enzyme is adenylyl cyclase, and it catalyzes the reaction of ATP to cyclic AMP. Because the adenylyl cyclase is an enzyme, once it's activated, it can convert many molecules of ATP into cyclic AMP. In this way, it serves to amplify the message by generating many molecules of cyclic AMP, or perhaps even just one of the one of the primary ligand on the outside of the cell. As with the phosphorylation cascade, it's not useful to have secondary messages hanging around long after their production and after the message has already been passed on. In this case, phosphodiesterase rapidly breaks down the cyclic AMP into AMP in order to shut down the message. Additionally, some G-protein systems use an inhibitory G-protein that prevents the activation of adenylyl cyclase. Thus, the cell has rapid and very fine control over the amounts of cyclic AMP produced. There are many, many pathways that involve cyclic AMP. One of these pathways helps to regulate chlorine and sodium ion balance in your cells, and unfortunately, this pathway has been exploited by the bacterium that causes cholera. The bacteria form a biofilm, or they sit next to each other, a biofilm on the surface of your intestines, and produce a toxin. The cholera toxin is an enzyme that changes a G protein, making it so it can't hydrolyze the GTP into GDP. As a result, the G protein is essentially stuck in its active form and continually activates the adenylyl cyclase, which in turn continues to produce cyclic AMP from ATP. The effects of this pathway is to cause the cell to expel chloride ions and not import sodium ions from the inside or the lumen of the small intestine. Having those extra ions in the lumen of the small intestine results in water leaving the cells through osmosis as the lumen becomes hyperosmotic relative to the surrounding cells. As a result, people with cholera have very severe diarrhea and may die from water loss. Understanding this pathway has led to some researchers developing nanoparticles that essentially act as decoys for the cholera toxin, basically capturing that toxin before it binds to and modifies the G protein. The effect of this treatment, at least on mice, is that the correct salt balance is maintained within the lumen of the intestine and water is retained by the cells. Maintaining ionic concentration gradients across the cell membrane is really common in cells. In the case of calcium ions, the cell uses ATP to actively pump Ca2+ ions out of the cytosol. The ions are pumped either out of the cell entirely or into the lumen of the endoplasmic reticulum or into the mitochondria. By maintaining very low levels of calcium ions in the cytosol, small changes in ion concentration can be detected. In fact, calcium is used as a secondary signaling molecule for many signaling pathways. So, how does calcium get released, and what's the signal for that release? In this pathway, the signal is received by a G protein coupled receptor, which activates the G protein, which then activates an enzyme called phospholipase C. Phospholipase C manages to cleave a phospholipid from the plasma membrane called PIP2, and it cleaves it into two molecules called IP3, or Inositol triphosphate, and DAG, diacylglycerol. IP3 is now a secondary messenger, and it diffuses to the endoplasmic reticulum, where it binds with a calcium channel. IP3 binding with the calcium channel is very similar to what we saw in the ion channel receptors earlier, and it has the same effect. It causes the calcium channel to open, allowing calcium ions to flow out of the endoplasmic reticulum and down their concentration gradient into the cytosol of the cell. One interesting aspect of this transduction pathway is that it divides in two. Remember when we looked at receptor tyrosine kinases, we showed that these phosphorylate tyrosine residues, which bind to various relay proteins, activating them to initiate different cellular responses. G protein coupled receptors, on the other hand, typically are only responsible for initiating one signaling pathway. This IP3 and calcium pathway, on the other hand, splits in two and allows for more than one effect on the cell. We've just now left how the cell receives its signal and how that signal is propagated through the cell until it reaches its target molecule. The response to a signal is often for the cell to produce more of a particular protein or proteins. It does this by turning a gene on through the use of a transcription factor. Transcription factors are molecules that bind to specific regions of DNA and cause the cell to transcribe more copies of that gene into mRNA. Ultimately then, the action of a transcription factor results in more copies of particular proteins. In addition to causing more copies to be made, some signaling pathways result in simple activation of proteins that already exist. In fact, this is often a very efficient way of regulating the activities of the cell. Instead of making proteins from scratch, existing proteins may simply be activated for use during a relatively short time period. There are several features of these signaling pathways that allow the cell to tightly control the amount of response that's generated from the original signal. The first method of regulation is simply that the signal can be amplified many times. Remember that signaling molecules may be relatively rare outside of the cell, and so it's often useful for those few molecules that find their mark on the cell to be able to elicit a very strong response in the cell. In the example shown here, a single molecule of epinephrine is bound to the G protein coupled receptor on the cell membrane. Once it has bound, it may cause the activation of 100 G protein molecules, and these will activate adenylyl cyclase. Since adenylyl cyclase is an enzyme, it can cause the formation of many cyclic AMP molecules. These activate many protein kinases in a phosphorylation cascade, and each step of this cascade generates more and more active molecules. Finally, the response in the cell is to convert glycogen into glucose 1-phosphate, and liver cells are able to do this such that one single ligand that's originally bound to the G protein coupled receptor that was able to cause the release of massive quantities of glucose 1-phosphate. You may have noticed as we've gone through these pathways that many of the molecules are common to more than one pathway. How does the cell keep these straight? In fact, consider what happens with hormones that are traveling through your bloodstream. They encounter all types of cells, but the only cells they have an impact on are their specific target cells. How did the cells achieve such specificity? The answer to this lies in the fact that each cell has a unique combination of receptors, transduction pathways, etc. Think about two cells that are receiving long-distance messages from the bloodstream. The only cells that will respond to such a message are those cells that have appropriate receptor molecules. Different types of cells have different receptor molecules and a different collection of proteins involved in the pathway. The differences in the pathways can result in very different responses to a message. Look at these 4 cells, for example. They're all receiving the same messenger molecule, but their response is different in each case because their unique signaling pathways has different proteins. In the last cell, the receptor protein is different, resulting in completely different set of transduction and response. In cell B, the signaling pathway splits, allowing this cell to maintain 2 distinct, simultaneous responses to the presence of the same signaling molecule. In cell C, there is crosstalk between these pathways, such that one pathway affects the other, perhaps by amplifying or blocking the signal in that pathway. There's a lot going on in a cell, and compared to the size of molecules, cells are really big with a really large volume. Molecules diffuse at a rate inversely proportional to their size, meaning that large molecules diffuse very slowly and small ones diffuse faster. However, even small molecules diffuse through the cytosol slowly. If the signaling pathway molecules were simply scattered around through the cytoplasm, it would simply take too long for them to diffuse through the cytoplasm and encounter the correct protein to interact with. Scaffolding proteins hold together different components of our pathway so that when a ligand is encountered, it can complete many parts of the signal transduction pathway. Additionally, the scaffolding protein ensures that all attached molecules are in the correct place. Another example of a scaffolding protein is this molecule called AKAP. This is a large complex of about 20 polypeptides and holds together the G protein, the enzyme adenylyl cyclase, the cyclic AMP, and even the molecule that recycles the cyclic AMP into AMP. Because molecular signals consist simply of molecules, it's important that these molecules don't persist longer than needed. The phosphodiesterase here breaks down the cyclic AMP into AMP, and we looked at a similar process where protein phosphatases dephosphorylate protein kinases in a phosphorylation cascade. One of the very useful functions of cellular communication is to communicate when the cell should die. This sort of programmed cell death is called apoptosis and may be the result of either internal or external signals. In the roundworm C. elegans, there are two genes, SED3 and SED4, which are inhibited by SED9. In other words, as long as SED9 is active, it represses or prevents the activity of SED2 and SED3. Once SED9 becomes inactive, it no longer has that repressive action, and SED2 and SED3 are free to initiate apoptosis or cell death. Sometimes, as in this picture, the signal for apoptosis comes from outside the cell, but often it comes from within. For example, during development, cells must die in order to form the correct form of our fingers and toes. Additionally, after new DNA is synthesized, molecules check to make sure the synthesis was carried out properly, and if errors that can't be fixed exist, the cell may signal for its own death.